Anagnostakis. Did I pronounce that correct?
Doesn't matter.
Okay, that means no. She has worked in genetics of various fungi, including those that cause corn smut disease and Dutch elm disease, and she has been working on chestnut blight caused by Cryphonectria parasitica since 1968. After completing basic studies with the fungus, she imported hypovirulence, that's virus-containing strains from France in 1972, and demonstrated that they could be used in the U.S. for biological control of the disease. She found the system of incompatibility in the fungus, which restricts the transfer of biocontrol virus, and developed the use of mixtures of strains for disease control. She and colleagues, Evelyn Havar, I'm sure I'm butchering that one too, found that strains produce very little or none. This has become the basics of her work on GMO trees with a gene for an enzyme that breaks down oxalate. She has worked on the ecology of the blight fungus and its control by hypovirulence and studies of virulence in the fungus and resistance in the trees. Sandra continues to work experimental station project on chestnut trees breeding to produce better timber and orchard trees. Seed orchards of improved chestnut timber trees have been established in Windsor and Griswold to provide seed for forest plantings. Test planting sites in the forest have been in place now for 16 years. Studies of superior orchard chestnut trees now include breeding orchard trees with Ozark Chinquapins for resistance to Asian Gall Wasp. I'm not going to tackle that scientific nomenclature. Seedlings from crosses of Ozark Chinquapins with Japanese chestnut and with the Chinese chestnut, Castanea Henry. And let's see, are being evaluated for their resistance to chestnut blight and Gall Wasp. Sandra is a past president and current secretary of the Northern Nut Growers Association, and sometimes judges the nut exhibit at the Pennsylvania Fair, and we're very, very honored to have you here. And I'm going to turn this over to you. Everybody needs to give her a good round of applause.
Thank you very much. Can we have the lights turned down so that people can see the slides? I'd like to start out by telling you that the Connecticut Agricultural Experiment Station was founded in 1875, and it was the first experiment station in the United States. So it was put in New Haven, Connecticut, totally independent of any college, and has continued to work just as a state organization ever since then. Now, the experiment station has had a long involvement with chestnut, and one of the first publications was ways of thinning your woodlot so you could make the best use of the lumber that was there and allow the rest of it to grow tall and use it in the future. And that publication, I think, was 1910. And there were many people at the experiment station over the years who worked on chestnut in various ways. And the involvement of Arthur Graves, who was a pathologist working in New York, but who had a home in Hamden, Connecticut, he started the work on chestnut breeding in 1930, and that work continues today. Graves and Donald Jones at the experiment station had many students who worked with them. One of them was Richard James, who now owns a very profitable nursery and did some wonderful work on breeding mountain laurel and has written a couple of nice books. But Richard James was the first person to really try and push forward the breeding program, which I took over when he retired. Now, I'm going to talk to you today about Chinquapins in Connecticut. This is possible because when Arthur Graves started his work in 1930, he started getting trees from the USDA and from many other people. And they were planted first on his family land and the experiment station farm. So I have all of this wonderful resource of every species of Castanea, I think, available to me so that when a new pest or disease comes in, I walk through these plantings and I see, okay, who's got the problem and who doesn't, and I have a clue about what kind of plant, what kind of tree might be used in a breeding program to improve resistance to whatever this new thing is. And that takes me to the Chinquapins. This is an Allegheny Chinquapin growing at our experimental farm in Hamden. And Allegheny Chinquapins are nice little bushes, and they are generally not very tall. They have clusters of burrs just like the Ozark that you're familiar with. This is the Chinese Chinquapin, Castanea henryi. The one on the left is a recent planting. There are not very many of these in the United States. One on the right was planted in 1930, one of Graves's early plantings of Chinquapin. That Castanea henryi was something that caught my attention when I was walking through a planting in Georgia at Callaway Gardens. We were looking at all of these trees that were badly infested with an insect called Gall Wasp, and I saw these trees that had no Gall Wasp, and I said to Jerry Payne, who was with me, "Oh, look, those Japanese chestnuts don't have Gall Wasp," and it turns out they were actually Chinese Chinquapin, which sparked my interest even more in Chinquapin. Here are some of the Ozarkenses that we have planted at the experiment station. They're very nice timber trees. We have trees from Oklahoma and from Arkansas, and I have a lot of dense plantings of them that I've used when I'm thinking about the resistance of trees to various diseases. And in this particular planting, it's been clear to me, there were, I don't know, about 80 trees in this planting, planted close together. And it's clear that from this kind of competition, that some of them are much more resistant to chestnut blight than others. And in my experience, the Ozark Chinquapins are more resistant to blight than American chestnuts. Now then there are all of these things. I say I have no idea what they are. I know they're Chinquapins, and I think they're in the Allegheny Chinquapin group, but they don't fit any of the standard criteria. And it was my annoyance at not being able to identify these that I convinced a young woman who's at the Arnold Arboretum. She's on a fellowship there. And she's a molecular biologist, and she likes to do DNA work. So she's now done a big study looking at all of the Allegheny Chinquapins and a bunch of Ozark Chinquapins up and down the East Coast, and has promised me that she will figure out how to separate them. This is work, of course, that's also being done by Hill Craddock and a student in Tennessee. They're using different kinds of DNA technology. So I think between those two people, we will finally figure out what all these funny-looking trees are that we know are Chinquapins. And it's because of this that I've always said that there are more Chinquapins in the United States than anybody has ever imagined. Now this is what Gall Wasp looks like. It's a small wasp, flies around and lays its eggs in the developing leaf and flower buds on chestnut trees. It was first imported into the United States by a chestnut grower in Georgia who went to Japan and saw some really nice trees, and so he put some cuttings in his pocket and brought them home without going through plant quarantine. And within about 10 years, he had eliminated his orchard of Chinese chestnut because of this insect that came in on those cuttings. You can see that these galls, these are leaf galls, and you can imagine that there's going to be not very much productive photosynthesis because usually the leaves die fairly quickly. Here's a map. The circle at the bottom is the place in Georgia where the insect was first imported by accident, and the spots are places where it was known. It got to Connecticut, this insect got to Connecticut in whatever I said, what did I say? 2011, and I knew it was coming. It's not just that the thing flies and gets around that way, but human beings, I always say human beings are just no good. Somebody will buy trees from a nursery where there's known gall wasp, and they think they can take care of it, but they can't. And so nursery stock is the way chestnut blight disease got passed around in the first place, and now this gall wasp is getting passed around the same way. So we've had gall wasp in Connecticut since 2011, and remember, I'd already seen those Chinese Chinquapins with no gall wasp galls. So I was overjoyed to find that the same could be true of Ozark Chinquapins. On the left is a European Chinquapin, a European chestnut rather, from Turkey. And you can see that it's got little lumps all over it. Most of that tree was dead in the spring. The tree on the right is an Ozark Chinquapin. There are no galls on that tree, and they're growing basically right next to each other. So this is my first indication that Ozark Chinquapin as a timber tree would be extremely valuable for crossing with any American chestnuts that we wanted to propagate as timber trees, because American chestnuts really suffer badly from gall wasp too. Not as badly as Europeans. The European chestnuts are devastated by it, but American chestnuts are pretty badly damaged as well. So I was concerned about American chestnuts and started crossing them with Ozark Chinquapins. But the other thing that I thought was a more immediate problem was the nut industry in the United States. And you haven't heard very much about it probably, but there are people making a living by growing and selling chestnuts. And the nut industry relies on quite a few established cultivars. And the problem is that because some of those cultivars have a lot of European chestnut in them, they're going to be in danger from gall wasp. The tree at the top here is colossal. That's a cultivar which is a European by Japanese hybrid. It's probably the most commonly planted cultivar in the United States. Cultivar, by the way, is a clone. You can only propagate it by grafting. And colossal is the cultivar grown mostly in Michigan. They grow others as well, but it's a very important cultivar. The one at the bottom is Eaton. Now, Eaton is one that was released by the Connecticut Experiment Station, and Dick James always said that he thought that was the best of all of the cultivars for taste of the nuts and form of the tree. And Eaton is it's Chinese by European by Japanese by American. So it has a lot of different kinds of genes in it. And so these are the two cultivars that I decided to include in this crossing with Ozark Chinquapin and Chinese Chinquapin. This is what the trees looked like when I was starting the project. There's colossal at the top, very badly damaged by galls. At the bottom is eaten. It's a little harder to see the galls on this. On the colossal, of course, the branches are dying from the infestation. There are some dead branches on the eaten, but not quite as many. My first process using these commercial cultivars was with Castanea henryi, the Chinese Chinquapin. And here are two of the results. I have a bunch of these planted. I have colossal by henryi and eaten by henryi. And what surprised me was that as the trees grew, yes, they did get galls. Not very many, but some. Now these are first generation hybrids. The funny thing was that a gall would develop and the branch would, a new bud would form and a branch would grow. And the next year another branch would start out. So the trees are sort of ignoring the galls and growing in spite of that. The crosses of colossal and eaten with Ozark Chinquapin had essentially the same pattern. The galls would form and the branch would grow anyway. Now that's last year's gall. Here's this year's gall. The leaves already died, but I wouldn't be surprised if a twig would grow out of that. And the result of this is that these trees go ahead and fruit. The galls are formed, the branches continue to grow, and the trees flower and produce burrs. Very often there's sort of an interruption in the branch in places where the galls are formed and really disrupting the growth of the tree. But the fact that they go ahead and make nuts means that a grower could use these and not be totally without a crop. There's a lot more work to do, but I think it pleased me to be able to tell people who said, oh, Chinquapins, you know, the nuts are too small to be of any use. And now I can say, well, you see, Chinquapins are of some use. They will save your trees from the Gall Wasp. So that's my story on Gall Wasp. Of course, there's a lot of other things that I've been involved in. These are, here's another Gall Wasp example. This is American Chestnut. It's a back cross, American by Japanese crossed again to American. And it's got a lot of Gall Wasp damage. And here's my Ozark Chinquapin crossed with Japanese, with flowering vigorously with no Gall Wasp damage at all. In fact, it's even better than the hybrids I have with other species. So I will be carrying on these crosses as much as I can. I am no longer a young person, and driving down to New Haven from Massachusetts takes a little effort. But I try to get down once a week and continue the work on the trees because they're not going to replace me because there's not enough money to do it. So I have to keep doing it. Now, one of the other thing I want to tell you is that I get a lot of samples. People will say, oh, I found this Chinquapin, or I found this chestnut, and they want to know what it is, and I can't tell them exactly, but I can guess pretty well from a leaf and twig sample. And the way I do that is to first just look, of course, at the forms of the twigs and what the leaves look like. but I have to look under the microscope. So I'm going to tell you a little Ozark Chinquapin story here. Here is one of our Ozark Chinquapins, and I call your attention to this is the lower leaf surface, and there are these little starfish hairs all over the lower leaf surface. So if I get a sample and it looks like it could be an American chestnut, and I look at the lower surface of the leaf, and there are little starfish hairs all over it, I know it's a Chinquapin. And from some other characteristics I can gather, it's an Ozarkensis. Here's an American chestnut. There are absolutely no leaf hairs on there. So it's very easy to tell Ozark Chinquapin from American chestnut. Now, this is a tree that was made famous by National Geographic and is also known as Hill Craddock's not dentata tree. Hill Craddock is a scientist in Tennessee at Chattanooga, and Hill had found this tree that he was pretty sure was an American chestnut, and they were going to use it in the breeding program, but there were some things about it that were not quite right. And the leaves looked right. There were no leaf hairs on the bottom of the leaf, but subsequently he and one of his students examined it more closely, and they found that around the edges of the leaf, there are these little hairs, he calls them ciliate hairs, and they are the clue that this is a different kind of Chinquapin. The tree has one nut per burr, and that was how Hill found out first that it wasn't an American chestnut. But this is not an Ozark Chinquapin. They renamed it Castanea alabamensis, and I got a leaf sample from the National Geographic tree that was supposed to be the surviving Ozark Chinquapin in the mountains, and that's what this is. And I think that tree is also not an Ozark Chinquapin, but Castanea alabamensis. So I look forward to some of these people who are doing the DNA studies actually finding out how many different kinds of Chinquapins there are. The Ozark, here's an Ozark Chinquapin. You see the little stellate hairs, and and also interesting to me is that many of them, clearly they have stellate hairs, and they're Ozark Chinquapins, but they also have these little ciliate hairs around the edge of the leaf. So what does that mean? And I don't know, but we need some good DNA technology to find out whether that's a hybrid between Ozark Chinquapin and alabamensis. Could be. There's so much to find out. And we could speculate forever, and until somebody gets some hard facts, got some genome analysis on these things, we won't know. But I know there are more species in the United States of native chestnuts, including Chinquapins, than anybody has speculated about yet. So, thank you. Is there time for a couple of questions, Steve?
Yes.
Okay. Oh, I'm stupefied, the entire audience. I'll be around the rest of the day to answer. Oh, well, there are some questions, yes.
Why do you think that the Gall Wasp don't like the Chinquapin versus the chestnut? What's the difference there?
The person who's thought about it the most, I think, is an entomologist in Kentucky, and she's done work on the insect. She thinks it's because the Chinquapins, both the Chinese Chinquapin and all of the American Chinquapins, formed their buds earlier than all of the chestnuts. And that somehow they're not attractive by the time the wasp is out flying. So I'm hoping that she'll get a little more information on that. That's the only clue we have so far. Yes?
I just picked up this dried leaf to look at it. You're talking about the hair like growth or whatever on the bottom of those leaves. How much do you need to enlarge that, see those?
Well, I can usually look at it with a hand lens, a 30 power hand lens, but you can feel them generally.
So
But
you feel it on a dried leaf? Or
yeah, even on a dried leaf. I'll check it later for you. And I have one of the things I just bought. It came from China, and I was thinking if I don't buy it now, there's going to be a tariff and it will never come. I just bought this wonderful little device which has a plug that plugs into my computer. It has a light, and it's a magnifying glass, and I can hover it over a leaf and look at these things in magnification from 40 to 1,000, and push a button and take a picture, which is how I got these pictures. It cost $44, and I couldn't resist. So if you're looking for something to use to take pictures, or you don't have a microscope, when I'm in New Haven, I can use my microscope that's still in the office, they still let me have. But I think this little device will keep me going for a few years if they don't let me have my microscope anymore. Yes?
Sandra, what about in the very uppermost of the range of the Chinquapins?
The young woman at the Arboretum and the people in Tennessee have got a lot of samples now. So I'm hoping, and they include some of the northern range, so I'm hoping that their work will clarify all of that.
They seem, the trees seem to flower according to ambient temperature and sunlight and all that. And the further north you go, the later they flower.
Yes, but and I don't know whether there's gall wasp in those places or not. We'll have to wait and see. Let's see, there was one other, yes?
What's the purpose of the hairs, or is there any defined purpose of the hairs between species and not having hairs?
I'll tell you something, a long time ago, when I was young and stupid, I asked one of my, the guy who was the director of the experiment station, we were someplace, and I asked him why something happened, and he said, because God made them that way. I don't know, I think of that, and think, oh, why did I say that? But I don't know if there's any purpose for those leaf hairs or not, but they're very convenient for identification. Yes?
Two questions. What is the limiting factor to the northern line of the range of the Chinquapin? And the other is the historic map of the pollen seems to be substantially bigger than any of the range that we've ever seen. What might be a...
I have no idea. The people who were out there looking at Chinquapins will have to tell you about the range, but that pollen map was developed by, I can't remember, somebody at a university, and it was wonderful. It was called, Fred, do you remember the name? Pollen something. Anyway, ask Fred, he'll remember.
I think he knows who it is. Well, here's the thing about the pollen. You can't tell Chinquapin pollen from any other
Right.
species of Castanea.
Right, yeah.
So
Well, the
that's
pollen,
that's a very large span, but it could be American Chestnut, Allegheny, it could be Ozark Chinquapin, and that's indistinguishable. You can't tell.
right, you can't tell the kinds of pollen. But the important thing was with this mapping system, it was on the computer, and you could go to it and you could say, any kind of oak. He did a whole bunch of different trees. And they did it by doing core samples in the earth and counting pollen grains. Fred probably knows this better. Ask Fred.
Well, the biggest issue here is that during times of extreme glaciation, pollen production is extremely low in some locations. And early palynologists made a big deal in places like Connecticut of coming up with all of these forest associations that really were based on long-distance pollen transport that had nothing to do with anything.
Right,
So
but
these
this program was so much fun because you click on it, click on chestnut, and it would sort of blossom. It starts out 500,000 years ago, and there's a little green dot, and then 100,000 and so forth, and the green dot gets bigger and bigger and shows you where all of the pollen is up until something like 1930, and then it gets smaller and smaller. And I had a young woman who was working on pollen studies someplace in the Northeast call me up and said she was very upset because she'd been looking at these soil samples, and the pollen, chestnut pollen, was increasing after the time when it had decreased, and it was coming up again in her samples in years by years. And I said, well, yes, you know how many Japanese and Chinese chestnuts have been planted in your area since then? And this is the thing. You can't tell one kind of pollen from another. And she was worried that all of the pollen stuff was totally wrong, but other chestnut trees produce pollen too.
Have you heard of the cruddy bark trees that some of the members of the American Chestnut Foundation in New York found? And what do you think's going on?
Fred Hibbert called them cruddy bark trees. Yes, I don't know. I suspected some bacterium or maybe a different fungus, just causing problems in the bark of the tree. And sometimes there are funny interactions with blight, and you get weird bark symptoms with that too.
Yeah, I think they're thinking that it's conferring some sort of protection because they're finding it on some really big trees.
Yeah.
That's what they're hoping, I think.
All of those surviving American chestnuts that were discovered, one here, they're all in the South. One in here, one in there, one in another state. And one tree has survived, and hundreds and hundreds died all around them. When people started finding those, we got samples from the cankers on, I think, most of those trees. And every one of them had a virus. They were not all the same virus. And they were not the same virus that I imported from Europe. The person who now knows the most about viruses, especially in Castanea and Cryphonectria, says that there are 4 distinct species of viruses, and they probably came from other fungi. So if other fungi were growing on the surface of the tree had a little more resistance, then there was a chance for the virus to pass from one kind of fungus to another. And that's probably how these American hypervirulent cultures, fungal strains, came up. And something may be happening like that with the cruddy bark trees. Okay, I will be here, so people can ask me questions later. If you have pollen questions, ask Fred.